Hi, everybody. This is Fatima here. I'm one of the senior TA candidates this year, and this is my video recording for the TA workshop. So the topic I've chosen to present to you guys is critical appraisal, or otherwise how to effectively critique a research paper. And this is important for most of us as researchers, but I feel it's also quite important as clinicians as well as as policymakers or people who are involved in generating clinical guidelines, etc. There is so much of published work in front of us nowadays, and I think it's important to understand why investigators publish their work. Of course, the main inspiration being to communicate their findings to their peers and to the population that they are aiming at with their studies, right? And of course, to make a difference in any aspect of clinical work, any positive change that they can bring in clinical practice or any aspect of public health. But there is also another individual aspect of why there is such a lot of publications out there right now that involves the investigators working towards their individual career growth, and also a somewhat misguided notion that the more published you are, the more credibility you get, at least on paper. And there is so much of papers out there that it's quite important for us to critically evaluate what we are reading. When we're doing our own study, we usually start off with a literature review. And if you know how to do a good critical appraisal, it helps us to filter out all the irrelevant studies and to reduce the information overload. It helps us to also see if the study in front of us is actually, can the results be applicable to the population that we are dealing with in our clinical practice or in our setting. And of course, peer review. Most of us are involved as peer reviewers in many journals, and this is an important part of our work as researchers. And I feel it's an important responsibility as well to know how to critically evaluate papers appropriately according to various checklists available, etc. Because I think it's a responsibility generally towards science and towards the population as well, so that the best works comes out, right? And something I think I have personally benefited from knowing how to do a critical appraisal is that I nowadays tend to focus on the content of the paper rather than looking at the journal name or the name of the big names of authors, in the author list. And that's something most of us should generally keep in mind, that big journals or big names don't imply always good methodology. They do have quite strict peer review process, etc. But sometimes it's possible that you find articles even in the biggest of journals that have fundamental flaws. So it's always important that you focus on the content of papers rather than, being fazed or be distracted by names, big names. It's also a way of self-improvement, because I feel the more you read and the more you critically evaluate papers, the more your own methodology improves, the more you try to be more honest with your methodology, you improve your work ethics, because you know that somebody is eventually going to be reading your work as well, and you want to put forth your best work for people to read. Your quality of work improves, your writing, presentation skills, etc. improve. And this is something that I have personally experienced as well. Before we move on to how to critically appraise papers, I think it's important to understand what types of scientific research papers are out there. You can have primary. These have not been previously published. They can be a primary analysis of data that was collected solely for the purpose of that particular study. Or it could be a secondary analysis of data that's already existing, data that's been collected for other purposes like survey data, or maybe data from any other study that you can utilize to answer a research question that's not been published before, not been answered before, right? And you can have observational studies, clinical trials, having primary or secondary analysis. Then you have secondary research papers where you use the data from studies that have been previously published. So our reviews, meta-analysis, etc., are sort of secondary because they use data that have already been published, right? So what are the questions that you ask when appraising a paper? I think it becomes a bit easier if you just split your mind into three compartments, and there are three major questions that you want to ask when you appraise a paper. Generally, one of the first things you look at when you are evaluating a study is what is the main research question. And if the authors have highlighted the main hypothesis or the main idea or the message they want to convey in the study, right? And this is often the first thing that attracts the readers towards the study, and often something we can identify from the title or from a brief reading of the abstract itself. And it's important to see if the message is clear or the main research question is stated clearly. If the study, in fact, has only one or more than one main research question, or if there is one research question and there may be subsidiary or additional specific questions. It's important to know or focus on this because sometimes it's possible that studies might include more than one main research question and therefore end up being very long, complex. And with complex methodologies often comes the risk of having invalid results. For example, if you look at the study of mother-to-child transmission of HIV, you can get multiple different questions that warrant individual papers on their own. For example, if breastfeeding is a risk factor for transmission, or if mode of delivery is a risk factor for transmission. If the authors decide to combine all of these questions in one paper, you can often imagine how complex it might be and how difficult it might be to actually extract the most relevant information from that. However, if you have papers, for example, if you have a study that looks at maternal nutrition during pregnancy and its influence on birth weight, you might find it more interesting if the authors even address specific parts of nutrition, for example, if fat intake or if carbohydrate or protein intake during pregnancy influences birth weight. And that's going to sort of appeal more to the readers. Or if you have a question like regarding hormone replacement therapy and the incidence of coronary heart disease in postmenopausal women, the authors also might be interested in knowing about the other major and the minor side effects of HRT in postmenopausal women. Although the CHD is the main research question. These particular sub-questions or specific questions cannot really be addressed in a paper of their own, right? But they make quite interesting additional features that might attract the readers towards these papers, right? So it's important to see how the authors have decided to address their question, and if they have gone for further specific questions or something like subsidiary questions, that might be okay. And the readers, it might be even interesting to read all those specific questions and see what the results are. But if they end up addressing too many main research questions in the paper, this might be difficult, and it might be difficult to even evaluate the validity of those results. It's also important to look if the research question is up to date or even relevant in the current setting. If they have done a competent review of literature, what has been recently published, and what is the gap of literature that this particular study is going to address. If they have defined the research question using the PICO format, because that is something that is universally understood by everybody. It's also important that we see if the hypothesis of the research question is actually testable. If you can actually measure the exposure or the outcome or the intervention and the outcome, and if it's possible to actually reach a conclusion about this research question. The second thing you want to look at is the methodology of the study, and this is quite a big topic on its own. I've just tried to briefly touch upon important points in the methodology. And of course, you begin with the study design. It's important to see if the study design stated by the author is accurate and if this design is appropriate for the research question that they want to address. also to see if it is actually the best design that is there for that particular research question. You know, you need to see if it's actually an experimental or an observation study, quantitative, qualitative. It's often quite useful if you can determine the timeline of events, because that gives a clue regarding the actual study design. And sometimes it's also possible that the authors don't really define a study design. So in order to make sense of the results, it's often important that you also determine the timeline of events, right? And you can find a lot of material online that sort of describes how these studies work. For example, if you look at cohort studies or and randomized trials as well, clinical trials as well, those are types of cohort studies. You start with individuals who are exposed to any particular factors. In clinical trials, it would be the intervention, and these would be individuals that don't have the outcome that is of interest, and they are followed up for over a period of time till and then you look at if how many of them develop the outcome and how many do not, and that is essentially what is cohort study, right? And in the case of case control study, you start off with individuals who have the particular outcome. You have individuals who have the outcome, and you have individuals who do not have the outcome. And usually the cases, there will be a subset of all the cases, and the controls will be a subset of the general population from which the cases came from. And you follow them backwards in time. So here the time factor is sort of from the outcome to the exposure, and you see if the exposure, how much of the individuals with the outcome had the exposure and without the outcome had the exposure. And that's how a case control generally works. And a cross-sectional is basically, you see at that particular point in time when you're conducting the study, how many individuals have the exposure and how many individuals have the outcome. And you try to make conclusions from that. Of course, the much less valid conclusions you'll be able to make from a cross-section when compared to a cohort study and a case control study, right? You can find various, checklists and flow sheets online published by many people that can give you a guide as to how to identify the study design in a particular study, right? Another thing often that you want to look at is the appropriateness of the study design. And by that, I mean, you need to look at the ethical concerns that are there, especially if you're looking at clinical trials. And if you think the particular study can be addressed by an observation study, then you want to see if ethically it was appropriate for the trial to be conducted in the first place. Often this might not be an issue because, of course, in order to even conduct a trial, they often have to go through an IRB process. But you would be surprised how many studies actually go through an IRB process, and you might still find ethical problems with the trial. Placebo use is an often debatable thing, and if you have a trial where placebo is being used, you need to actually look into it more deeply and see if it was a valid use. And if you have studies that have used designs that have been previously published, and you find a justifiable reason for the authors to use the same design to answer the same research question, this is also a problematic thing. If you are doing a same design that has been done before, there should be a valid justification for repeating the design. Probably the population is different, the setting is different, something that would explain why you're doing it again. And is there a better design? Often, one of the things that the peer reviewers often look at is if this particular study was the best to answer this research question in terms of cost and feasibility. The next thing you look at is if the study population and if the way the sample was selected is appropriately mentioned, right? And if the whole process was appropriate for that particular research question and for the study design, right? And the famous diagram that all of us are familiar with. You have the population, the target population, the accessible population, from which you have the sample that is selected using the sampling method. right? And one example that I often like to state, for example, the population is all the pregnant women in the country, just an example. The target population would be all the pregnant women that have an antenatal care registered in the hospital that I want to look at. And my accessible population might be all the pregnant women registered in the hospital, but under one particular team, because I have access to only those patients under that particular team. And sampling frame is usually a list of all the patients, the entire population or the accessible population that is available to you. This is often important in public health studies where you need to have the entire sampling frame and where you're going to be doing a probabilistic random sampling method, right? And then you do a sampling method like probabilistic or non-probabilistic in order to get your sample out. And often the sample is also defined by certain inclusion-exclusion criteria. For example, only pregnant women who are registered in the first trimester with pre-existing hypothyroidism, and that's the inclusion or the exclusion criteria that you apply to it. Okay, this is just one of the examples. And it's important that all of these parameters are quite, specifically mentioned in the methodology section so that the person who is reading it can actually decide if the results of the study is applicable to the population that they are concerned with, or their own population. The comparison groups or the study groups, often the next thing that needs to be clearly defined are the study groups. In observational studies, exposed, unexposed, in clinical trials, the intervention group, the control group, how they were selected and the randomization process. In case control studies, the cases and how the controls were selected. Are they representative of the population that you want to extrapolate the results to? In the case of observational studies, any steps have been taken to account for confounding, for example, matching. That's something in case control studies also that is often done, where the cases and controls are matched to each other. This is done in an attempt to control for confounding. Has any of these processes, then it's important to look at that as well. In methodology of, when you're looking at a methodology, the way I look at it is, have they given enough information in their manuscript that allows somebody who is not aware of anything about the study, who comes in and reads the paper and would be able to replicate the study exactly and get the exact same results. That's the idea behind writing a good methodology section, right? The next thing you want to look at is the variables, the various definitions, and then how the variables have been measured. The variables can be either clinical or lab or based on questionnaire data that are taken from patients. And you want to have a clear definition for all of these variables so that, it's enough, like I said, methodology where the study can be replicated. And if in the case of measurement, it's also important for the authors to mention the measurement tool that they have used, and also to highlight the validity and the accuracy of the measurement tool that they have used. And if the same measurement has been utilized for everybody in the study, and if there was a requirement for taking blinded measurements in order to reduce bias. For example, if our exposure is body mass index, right? One way of reporting it would be to write the exposure is the body mass index calculated as so-and-so of more than 50 in the first antenatal booking visit, etc. And that would be a very way of clearly defining what the exposure is. And then how it was measured, would be something that can be written as, for example, height and weight measured using standard hospital scales by clinical nursing staff and manually entered, etc. So it's important that the authors clearly define the various variables, not only the exposure and the outcome, but also the other variables that are included that might be needed for the analysis. The choice of variables is also quite important, and it should be relevant for the particular research question that they want to address. The independent variable would be the exposure variable would be the main exposure answering the given hypothesis. And clinical trials, often this is the intervention or the drug, the new drug that is being tested. Often you see that in case control studies, because the timeline moves from the outcome to exposures, it has an advantage of studying multiple exposures at one time as well. Then you have the dependent variable, which is the outcome. Again, depending on the number of questions that are there, you can have a primary outcome and multiple secondary outcomes. Another thing that you want to look at is if the outcome used is a surrogate, right? And for example, if the study wants to look at mortality, but instead of looking at mortality because of whatever reason, they look at a particular event before that and decide that that's going to be the outcome. And they're going to extrapolate that or make the inference from this association and say that, okay, fine, if the exposure is related to this particular outcome, then it's most likely that it's related to mortality as well. And if that's the case, then it's important that the authors mention how the surrogate is linked to the intended outcome. So how this middle variable is linked to mortality as well. And if this has been validated and if this has been proven before in literature. Often, of course, in clinical trials, probably not so much of an issue. But even then, it's important to collect variables, demographic variables, and to get information about all possible confounders and effect modifiers. It's much more important, of course, in observational studies. And also to see if the authors have defined the proper follow-up time, because you need to have a follow-up time enough so that the outcomes can happen. Or for example, if it's a study on side effects, you need to give enough time so that you can observe side effects. If you have a limited time or a reduced time, and the authors conclude that there are no side effects, this might be something that you can question. And if they have data of those participants or subjects that are lost to follow-up, and this is something that might introduce bias into the results. And it's important to see if this has been mentioned or appropriately addressed by the authors as well. Sample size, again, it becomes important to provide a rationale for the sample size. In any analytical study where you're going to be comparing two groups, it's often important that the sample size calculation and the literature review that has been done in order to get to the particular sample size, this needs to be mentioned quite clearly, or the numbers or the figures that have been used to reach the sample size. And it's also important for us to judge if the sample size that have been given is enough to prove the hypothesis mentioned by the authors. And then coming to statistical analysis, of course, this is another big section on its own. But it's important to look at the various variables that have been used and to see if they have been defined properly as either categorical or continuous, and if they have been classified appropriately. And if continuous variables have been categorized, what is the rationale behind the categorization of these variables? And then if the appropriate tests that have been used are correct based on the type of dependent and the independent variable. If there was any requirement for any adjusted analysis, and most importantly, how the missing data has been handled. And you would expect, these minimum things to be mentioned in the statistical analysis as well. Presentation of data. This is something that the authors need to be really careful about. For example, when you're generating the tables, it's quite a tedious work, and often this is the place where a lot of mistakes do come in. And I think as reviewers, or when you appraise the paper, and these are areas where you do need to focus on, because often times after publication, so many times you see papers where there were errors in the tables or in the results presentations, and this was completely not seen by any of the peer reviewers, right? You need to see if the numbers in the table actually make sense and do they add up accurately. If there are percentages that are mentioned in the table, do you actually know what are the denominators for the percentages, because it's possible that the authors just mention percentage, but you don't really know it's a proportion of what or a percentage of what, and you have the concept of a row percentage or a column percentage. If you are dealing with cohort studies, you often have the exposure groups or the comparison groups in the columns, and you generally want to have column percentages. And this is something you need to look at the table and try to figure out if this has been clearly mentioned by the authors. If they have presenting graphs, are the X and Y axes appropriately marked? Do they begin from zero? Because often you have scenarios where they have just taken a snapshot of the graph and presented, and they don't begin from zero, so you just get a snapshot, and the conclusions might be completely different from when you see the overall graph. Are the scales that are used reasonable to represent what happens in reality, or have they been just blown up to exaggerate the difference? And this is something that you see quite commonly in studies as well, so that they can show that there is a big difference when in reality there is not. For example, you look at this particular graph. In this section, on the right side, you see that the scale starts from 0 up to 3.5. And you see that every year the bars are almost the same. But on the left side, if you just focus from between 3 to 3.5%, you see the difference, right? So if the authors use this particular graph, you feel that there is a huge difference between the years, when in reality, it is not that significant. This particular figure, if you see, the numbers are quite different for abortions and for cancer screening. And the authors have not defined the y-axis over here. So it's just randomly, they've just put the lines, which give a very wrong impression. You get the idea that abortions are increasing dramatically, whereas cancer screening is decreasing. When in reality, the number of cancer screening is really more, still much more than the abortion services that are there in 2013 as well. The trend might be okay, but then claiming that the abortions are, taking up more resources than the cancer, that would be something that's false. And that's the impression that you get from this particular figure, because less number of people tend to focus on the numbers, right? Another common mistake you see is pie charts where the segments don't amount to 100%. So here you can see there is 50% marked, 60%, and then 38% marked. And they don't form, it's not a pie chart. All the segments should add up to 100%. So it's important to see how the results have been presented as well, right? The last part that you want to focus on is the validity of the results. And more importantly, is the interpretation of the authors valid? And if you can generalize these results to your population. The results section, it's important to see if the estimates that they have presented, they're appropriate for that particular study design, and if the estimates have been presented with confidence interval to show the precision of the results. For example, if it is a binary variable, for example, disease occurrence, yes, no, you have various ways you can show the results. For example, risk rate, prevalence, odds, proportions. And then you can show the measure of effect or the measure of association using various estimates like risk ratios, rate ratios, hazard ratios, odds ratios, risk difference. And you can go one step ahead and show measures of impact of that particular exposure on the population as such. So that's showing population attributable, or this would in fact be showing the effect of the exposure on the outcome in the population generally, and not only in the sample. And that would be the measures of impact. And it's important to see if these measures have been appropriately used. So if there are odds, is how the measure of occurrence has been measured, then they should be using odds ratio appropriately. It's sometimes also possible that the authors initially state the risk, and then when it comes to association, they use odds ratio, which is also fine. But preferably, if you're doing risks, you need to show the risk ratio. If you're doing rate, then definitely you need to show the rate ratio, and in the case of survival analysis, hazard ratios. Um, in certain study designs, for example, in case control study, you wouldn't be doing incidence risk and incidence rate, and you would be only using the odds and the odds ratio. And that's, of course, a completely different discussion on its own. But that's just one of the examples I wanted to mention regarding using the appropriate estimates for the particular study design. In clinical trials, it's possible you might have continuous variables as well, and there it's important to look at the distribution of the variables and if mean and standard deviation is more appropriate, or if the median and the interquartile range is more appropriate. These are just some examples of how the results can be displayed or results are presented. The next important thing is how the authors choose to interpret the results. And here, the words used to describe the results are important. For example, risk, rate, odds often are used interchangeably, which is a problem because each of those definitions are different. And often, something that's very commonly done is the interpretation in terms of the evidence for or against the null hypothesis. And this is to see if what is being observed is more than mere chance. And, again, this is something, and how do you define as chance is having the p of at 0.05, etc. So in this particular sort of interpretation, it's important you see if they have looked at the strength of the association, and not only the strength or the effect, the size of the effect, but also if they have shown the confidence interval at the width of the confidence interval to tell us also the precision, right? It's necessary to see if the authors have accounted for any confounding, and if these have been the interpretation is appropriate after accounting for the confounding. There are so many types of bias that can get into a study, and I think it's the limitations and strength of the study, the section in discussion. I think this is a very important part where we can actually know if the authors are being transparent or honest about their study design and their methodology. And it's important that we interpret the results of the study in looking at the strengths and limitations section as well, because then we can get an idea about the possible bias that is there. And at times, it's also possible that the authors have not even thought of this particular bias that could have been included in their methodology. And it's important then as peer reviewers to highlight that this is a possible bias that could have been there, and request the authors to address it in their strengths and limitations. So it's the idea is that after looking at all of this, we need to see if the results are actually believable, and if the interpretation of the authors are not far-fetched from the actual results that are there, right? And this is a sort of flowchart that I had prepared previously just to show the types of bias. And I think when you take up the responsibility of doing a critical appraisal, it's important to have a good background knowledge of the different types of bias that you can incur, be it in clinical trials or be it in observational studies. And to, be able to think critically, looking at the setting of the study and the methodology, and to see what sort of bias can be introduced into those particular studies, and if the authors have taken the necessary steps to combat those bias. It's not only that they have taken the steps, they should also be able to highlight it in their manuscript, and so that the readers are aware that it has been done, right? And another part in the discussion section that is quite important is to see the similarity of the results with the other studies that have been published globally, right? Often the authors also need to mention if there are any differences between the results in their study and what has been globally published, what could be the possible differences, reason for the difference in the results, and to also provide information about how the gap in literature has been addressed in their study, right? And if the conclusions are actually possible, because if what the authors are claiming in their particular study is not even biologically possible, then that is a problem, right? So this often takes up the discussion section of the study, and it's important that we give this portion a good read so that we can know how the results of the study actually sit with what has been published previously. And this also helps us to see if the results of this particular study can be applied to our population as well, to the setting that you're working in, to the resources that are available, and generally to assess the possibility that if this particular study was done in your population, would you have got the similar results, right? And this is quite important when you are sitting in a committee generating clinical guidelines for a particular setting. It becomes important to choose studies or to base your guidelines on literature that are based on studies that are done in population that are quite similar to yours, the population or the setting that are similar to yours, so that the results are valid in the particular population or setting. So in summary, the three main questions that you look at when you do a critical appraisal is the three main questions are: what is the research question? Main research question: is the methodology appropriate? And are the results valid and generalizable? And there are many checklists and resources available that are used generally by peer reviewers. You have the CASP checklist, which is quite famous and freely available online for every type of study. You also have the stroke checklist for observational studies. This is basically to give an idea about how to write observation studies, and that should be included in the reports. I often use the stroke checklist to write my articles as well as to do peer review or a critically appraisal article. You have the CONSORT checklist for RCTs, and you have the PRISMA for systematic reviews as well. And remember, when you're doing a critical appraisal, I think it's important to remember that it's often a give and take. When you enter this world of publications, people are going to be reading your work and people are going to be critiquing your work, right? It's easy to be on the other side and give feedback to somebody, but it's often difficult to receive feedback. And it's always necessary that we remember that it's always a give and take, and we should be open to criticism, and we should be open to making positive changes in our study. So that's it. Thank you for listening, everybody.